Hello, hello, biology. All right, this is the first video on cell transport. Here's the bell work for the day. Again, 22nd was 7th hour. 21st was 3rd hour. So 3rd hour did this a day earlier. So I wanted to mention that. This actually starts off with 2 videos, which was a pretty good chunk of the hour. We watched a crash course video, which should be linked in the description below, and then we watched a Amoeba Sisters and Amoeba Sisters video after that, which should also be linked below. And I should have the titles for those so you know which one's which. We watched both of those and then started on the notes. So there's not a lot of notes. This video is going to be pretty short because it's mainly just kind of reaffirming a lot of the stuff that's in those. So that said, let's get started. Okay, this is the graphic from, or a graphic from the Amoeba Sisters video that I wanted to specifically point out because I've had a lot of people get confused about facilitated diffusion. Facilitated diffusion and simple diffusion are both passive transport. This is not active. This is still passive. The difference between these two is this just goes all the time. This can go through the phospholipid bilayer. Okay, and this is mostly just gases. This actually requires a channel. Okay, because for things like liquid or water, that cannot go through the phospholipid bilayer because it's water repellent. So they have to have some kind of protein channel in order to actually get through. So transport proteins is the difference. This just goes straight through the bilayer without any help. This has to have a transfer protein. I can't read backwards. Transport protein. Okay, so that's the main difference between the two. But they are both passive transport, which means neither one of them moves against the concentration gradient. They both move with it. Okay, now that I've stepped on that soapbox for a little while, now we'll go back to the actual notes. Okay, cell transport is broken up into 2 different parts because it's 2 days. It may end up in 3. I'll have a better idea of that after class tomorrow. But this is part 1. Once again, this first part is a piece of background information, what exactly a boundary is. The main thing for this is that the cell membrane is the barrier or boundary for the cell. Okay? It is semi-permeable. Semi means partially permeable. The best way to think of permeable is pass-throughable. It's how easy it is to get through something. So a semi-permeable barrier or a selectively permeable barrier is a barrier that will allow some things to come through and will not allow other things to come through. Think of it like the border patrol for the cell. Okay, if you look sketchy or if you're a sketchy person, maybe you're on a wanted list, they're not going to let you come in. But most people will be able to do just fine. And yeah, it regulates what comes in and what goes out of the cell. Here's just a diagram. This is the diagram from your notes. So this image is just straight out of the notes. Notice that it has lipids. It's got the phosphate heads, the lipid tails. We've got proteins embedded all over the place. Carbohydrate chains that are usually going to act as the receptors that were talked about in the video. But these proteins are usually going to function as the channels that were described in those videos. Oh my gosh, I'm blanking for the second kind of diffusion, facilitated diffusion. These are the kinds of channels that are going to allow for facilitated diffusion. Okay, then we have this. Now, my question is, what is the word that goes here? Technically, it is proteins. I would prefer if you actually said transport proteins. But there are several others embedded in there, so that's why this is saying proteins. But bear in mind that specifically when we're talking about transport, we're talking about, or cell transport, we're talking about transport proteins that are within that cell barrier. Okay, the structure of the cell membrane is what regulates what is allowed to come in or go out. If certain proteins are not present, certain channels are not present, then nothing can pass through that membrane, or that particular material can't. For example, water. They mentioned it has a very specific transport protein that allows water to move through in and out of the cell. Well, if that protein is missing, then that cell is going to have a lot more difficulty moving water into or out of it. So the structure can have a serious limitation on what that cell is able to move around. And that is it for the notes. This was the last assignment for the hour. What I want you guys to do, for those of you who are not in class, on a sheet of white paper, I want you to sketch this out to the best of your ability. And I'm going to go turn the lights on real quick so you can get a better look at it. Hopefully that's a little clearer. Yeah, that's a little better. Okay, and I am probably going to upload an image of this into Classroom so that you can pull it up and look at it maybe a little better. No promises on that, though. I'm still on the fence. But you need to sketch this out, including all the spikes and hairs and things coming off. Be specific. Take your time. Do it well. And then you'll need to actually label. I'm going to point these out. This is an A, B, F, G, H, I, C, and E. Because I know those are somewhat hard to spot. Then we have A, B, C, G, H. I is pointing at these two things right here. F and E. And C, by the way, is pointing at this green thing right here. Okay, so that's to kind of help you with your labeling. Take your time. Do it neatly. Each one of these is one of these. Okay, so don't just draw a bunch of squiggles all over the place. Take time. Do it well. Make it look neat. And then I'm going to have you actually insert that into the notes. The next video, I will show you where I want you to insert it. That is it for this video. I will see you guys in class. Have a wonderful night.